Welcome back, cell biologists. So we've been discussing transcription of our RNA molecules. We've talked about how RNA polymerase binds to a promoter. We've talked about how it initiates that transcription of RNA. And now let's talk about how it elongates, or how it keeps that RNA molecule growing longer. So this is our RNA polymerase. Here's our DNA, and here's our RNA. On this picture, the purple RNA polymerase, your blue RNA, and your red RNA and your blue DNA. So as elongation is happening, we are building a longer and longer RNA polymerase molecule. Your RNA polymerase is moving along DNA and using that template of DNA to know what nucleotide to add to your RNA molecule. The beta subunit of RNA polymerase has a little pincer, and that pincer is able to bind DNA, kind of open it up, push it into the active site, and that allows for things like new nucleotides to come in to be added to the growing chain. It allows things like the completed RNA, the completed DNA to exit after transcription has occurred on that segment. RNA is always elongated in the 5' to 3' direction. That means that the 5' end is where it started, and the 3' end is where the new nucleotides are added to it. It's not going to come back and elongate from here. It's always in the 5' to 3' direction. As RNA polymerase is moving along the DNA strand, it's going to be unwinding the DNA ahead of it. And then as DNA is exiting, DNA is being rewound. And you can imagine that if you were to just grab two things that are twisted together, two cords, and just pull them apart, try to open it, you might end up with some supercoiling, some really intense coils happening here, maybe even tangling or breaking the strands. We don't want that to occur. So topoisomerase is going to be an enzyme that comes in here and relaxes that DNA strand to allow it to coil and uncoil correctly without worrying about breakage or knotting up your DNA strand. RNA polymerase is able to proofread what it's working. It's kind of a weak proofreader. It's not great, but it's okay if it's not great, because RNA molecules are kind of short-term molecules anyway. So if you have an error in RNA molecule production, it might make a temporary impact, but it's not long-term going to affect the ability of that cell to survive or to reproduce. So it's not very critical compared to the errors in like DNA replication. There's two ways that we can do proofreading. One is that RNA polymerase can go forward or backward. That means it can add or remove a nucleotide. So if it's going along adding nucleotides, it gets to a strand, it has just added the wrong nucleotide. When it does that, it kind of pauses. And when it pauses at that spot, it's more likely that that reverse action, that removal is going to happen. So that the incorrectly added nucleotide is removed. Another thing is that if the incorrect nucleotide has already been incorporated, the polymerase can backtrack. RNA backtracking means that the polymerase will back up a little bit. It'll take out that incorrect nucleotide and also the piece of RNA previous to it, and then continue on with transcription, adding the correct nucleotides that time. Then eventually RNA polymerase is going to get to the terminator sequence. And when it gets to the terminator sequence, it's going to terminate or end the RNA synthesis. And so that is the end of transcription. There's two different kinds of ways that termination can occur. One is using rho factor, and one is not using rho factor. That rho should say RHO, rho. So the first one is without a rho factor. If this molecule of RNA does not need to have a rho factor, the RNA is going to include a GC-rich sequence. That means that there's lots of G's and C's being produced in this segment of RNA, and it's going to form this hairpin loop as those G's and C's interact with each other. Then downstream of that GC subunit of that GC sequence is a segment that's full of U's. And all of these U's and A's will have a slightly weaker bond than other nucleotides comparatively. And those slightly weaker bonds allow this RNA molecule to kind of break its interaction with the DNA and release the RNA. It's able to float away. If your RNA molecule doesn't have that GC-rich area, it won't form a hairpin. It'll need a rho factor for termination. And in this picture, this red stuff is your rho factor. Rho factors are going to bind to specific termination sequences at the 3' end of your gene. These termination sequences are typically around 50 to 90 base pairs long. Rho factor is an ATP-dependent unwinding enzyme. That means that it takes the energy from ATP and uses it to unwind or to take the RNA and the DNA and separate them from each other. So in this process, RNA is freed and able to leave from DNA. So you have terminated this transcription of that gene. All right, so we've talked about our 4 steps: binding, initiation, elongation, and termination. Next, we're going to talk about how eukaryotic cells have slightly different RNA polymerase, the slightly different structures, the other differences in transcription like RNA processing, some interesting things still to talk about. So I'll see you in a little bit. Let me know if you have any questions. Bye.